Hello friends, welcome to another exciting video from my everyday science. We are going to continue with our recombinant DNA technology. That is where we left off the other time. So with recombinant DNA technology today, we are going to look at the detailed process of what actually goes on. So we'll be looking at the steps in recombinant DNA technology. I am Mr. Kutam Gabriel Pizasso, and I'll be taking us through today's presentation. So let's have a quick recap from what we learned last week. So last week we actually talked about our restriction enzymes. We said these enzymes are also known as molecular scissors, and what they do is that they cut the specific DNA sequences. So be it our gene of interest or our plasmid DNA vectors, these restriction enzymes will cut these sequences for us before we bring them together to have our recombinant DNA. We also talked about vectors, and we said these vectors are vehicles for transporting DNA sequences into host cells, because when we have this recombinant DNA, we have to multiply it, we have to let it replicate so that we can have sufficient amounts to analyze. And this vector helps us to actually transport them into our host cells, which we mostly use bacteria. So we would be moving straight off to the steps in recombinant DNA technology. So first, we obtain our gene of interest, which is known as isolation. We insert the desired gene into a host cell for replication, and most of the times for our host cells we use bacterial cells. Now we extract the product of these recombinant DNAs. So when we have these recombinant DNAs and then we insert them into a living cell or a host, they replicate. So when they replicate, we get the products from their replication, and we actually analyze these products. Then after they replicate, we have to select or screen to identify which cell was cloned. So let's do isolation of a gene of interest. Now the genome of an organism contains several genes. Now for our recombinant DNA technology, there is a particular gene that we are interested in cloning, that we are interested in making copies of. So that gene is what we are going to cut out of the sequence. So we will take the organism's genome, we analyze the genome, and then we cut those parts, which is our next step. We gene location and isolation by means of restriction enzymes. We locate these genes and then we isolate them with the help of restriction enzymes. We talked more about that in the other video. So these restriction enzymes are of two types, and then these types are based on the types of cuts that they make. So depending on the cuts that they make, DNA restriction enzymes are of two types, and these are cuts with sticky ends. So some DNA cuts sequences with sticky ends, and others, after cutting, leave blunt ends. So this is what the sticky ends actually means. So when we say our DNA has a sticky end, this is how it looks like, and this is how DNA molecules with blunt ends actually look like. Now most of the times we do prefer the enzymes, restriction enzymes, that cut and then give us sticky ends to those that give us blunt ends. Now this is because those with sticky ends easily bond together through weak hydrogen bonds. So we can have an example of the vector. We can have our vector plasmid DNA, which is also cut with these restriction enzymes. So we use these restriction enzymes to get our gene of interest, and likewise to cut sequences into our plasmid DNA vector. Now after cutting our DNA vector and then getting our gene of interest, we have to bring these two together. We have to join them for them to form a bond, and this is done by the DNA ligase. So in our next slide we are going to talk more about our ligases. Now when we join this DNA fragment, that is our required gene and then the plasmid DNA together, then we have what is called a recombinant DNA. Now onto ligases. So these are enzymes that facilitate the joining of DNA strands together. So after we've gotten our gene of interest and then we've cut sequences into our plasmid DNA vector, what we do is that we bring these two together, and these ligases help us to ligate these enzymes, these DNA together. They do so by forming phosphodiester bonds. Now we know that nucleotides, successive nucleotides, bond together through phosphodiester bonds. So these ligases are going to help so that there will be production of these phosphodiester bonds in a haste. So they catalyze the formation. That means they speed up the formation of our phosphodiester bonds. We can have an example of the ligases as the E. coli DNA ligase, and this does ligate sticky ends, but rather it can't ligate DNA fragments with blunt ends, but it can do so under one condition, and that is molecular crowding with polyethylene glycol. And then also it can't efficiently join RNA molecules to DNA. So let's look at how to now insert our recombinant DNA into our host cell, for which most of the times we use bacterial cells. Now for our insertion, there are different types. We can have artificial methods of inserting, and likewise we have natural methods of inserting. So for artificial methods, we have electroporation. As the name suggests, electroporation, that means it uses an electric field or a power source, and then poration to create pores in the walls of the cell. And when these walls are created, the cell wall breaks, and then we can now fuse our recombinant DNA and our bacterial host cell. Likewise, we have protoplasm fusion, and with this we are going to apply enzymes, and these enzymes are going to remove the walls of the bacterial cells, and then with polyethylene glycol we fuse the cell, and then the DNA recombinant or the recombinant DNA. We also have injection, which we can do by using a gene gun or a micro injection. Now for our natural methods, we have transformation. Now with transformation, we treat these bacterial cells with calcium chloride. Now what these calcium chlorides do is that they influence the cell to a state known as competence, and this competence makes the cell able to receive our plasmid DNA. So after influencing this competence, we add the DNA to the bacterial cell, and then we apply heat, and then the plasmid DNA is now fused into our bacterial cells and then begins to replicate. Now we have to note this: when phage is used as the vector, there are different types of vector, and then you can choose from any of them. But when we choose a phage as our vector, because these phages are bacterial infecting cells or bacterial infecting viruses, they do not need to be transformed. They can just affect or infect the bacteria easily. So there will be no need to use this transformation method when a phage is used as a vector, since they infect bacterial cells already. Now let's look at how to select or how to screen our cells to know if they actually take up the recombinant DNA that we introduced into these cells. Now, as a matter of fact, we know that not all these cells can incorporate the recombinant DNA. That is, not all of them can take up this DNA because of some experimental errors. And because of that, most of the times we use a lot of cells, a lot of bacterial cells, and afterwards we select the ones that actually took up these plasmid vectors. So the bacterial cells are plated, and when they are plated, they are treated with antibiotic cells. Now, one thing is that which we are to note is that these plasmid vectors make the bacterial cells resistant to antibiotics. Now, the main function of antibiotics is to resist or inhibit the growth of bacteria, but because these plasmid vectors that we introduced into the bacterial cells make them resistant to antibiotics, that means when we apply the bacterial cells with antibiotics, the ones which took up the recombinant DNA are actually going to survive and are not going to be affected by us antibiotics. Now, for a summary of the whole process, we have our gene donor or the organism's genome. This is the organism's genome, and then we isolate a gene of interest. So this is our isolated gene of interest. From the structure, we can see that it has a sticky end, and then we come to our plasmid donor, which is a bacterial cell. We take the plasmid out, and then we apply the restriction enzyme as well to the plasmid vector. Now, what happens is that we ligate these two together. And then we get our recombinant DNA molecule. And then we introduce our recombinant DNA molecule into our host cell. But this time we are using the transformation. So when it's in our host cell, it replicates, and then we can now take our products and then use for whatever research or whatever purpose that we would like to use it for. So that's it, guys. I hope this video actually helps you, and if it did, please don't forget to hit the subscribe button, like, and also let us know your views. Let me know your views in the comment section. Don't forget to share the video to your friends also, and as well, thank you.